Great, thanks so much. Yeah, thanks so much for organizing this, the opportunity to talk about these issues with all of you. I'm actually looking at the title right now, and I'm not actually that happy with the title. I wrote this. I'm not blaming my co-authors here, but because our central point isn't so much to question the notion that the proximate ultimate distinction isn't somehow theoretical bedrock. But what we want to do is just bring to the attention, take this opportunity to bring to the attention of philosophers of biology things that are happening in biology that they might not be aware of, and cover things that are of importance for biological practice, which in the end is of interest to us as authors because we're biologists. This talk actually came out because the five of us were working on a review on the cause of a very widespread morphological pattern in plant hydraulic systems. And we come from a variety of different perspectives. And what we came up against were very different notions about what constitutes a good explanation in our field. And there's a lot of heterogeneity in the group. So I'm trying to reflect a consensus, but I take responsibility for any misrepresentation here. What I want to do is provide some examples from our field, which is this field of plant hydraulics. And so, as Özlem alluded to, plants, especially vascular plants, are very important. One big reason is that, I don't know if you can see my little arrow here on the left, but they represent the bulk of organismal carbon on land. So this is a graph showing the relative values in gigatons of different organisms. And you can see plants here, like the vast bulk of this. So animals aren't neither cool nor interesting, right? They're totally trivial in terms of organismal carbon biomass. And this carbon is fixed via photosynthesis, which involves the transport of vast amounts of water. And plants do this water transport through conduits in the wood under negative pressure. So the water's in a metastable state. So the water's under tension. It should, by all accounts, if it weren't for the very, very particular conditions of the xylem, would be turning into gas. So very unusual situation in nature and extremely hard to study. It's a really, really challenging experimental situation. And the field that studies this is variously called xylem physiology, plant hydraulics. And we're interested in plant hydraulics because of the importance of plant water conduction. What plant hydraulics tries to do is understand how hydraulic function emerges from variation in structure. So here's a cross-section here on the left of a eucalyptus twig. And you can see some of the tubes that transport the water. They're these round open spaces, or at least some of them are the tubes that transport water. And as Sabina and others have referenced as well, water transport's important. Well, they mentioned these important aspects of plants, and water transport is intimately involved in these in underlying terrestrial primary productivity, carbon storage, carbon fixation, right, is the essence of our food production, and failure of the hydraulic system, so breakage of the convective stream, is involved in ongoing events of forest mortality worldwide, which leads to these vast losses of ecosystem services. So you can see these dying trees across the landscape. And again, breakage of the hydraulic system is involved, at least in some way, not necessarily the only cause, but involved in these systems. So we're interested in making these structure function understanding the structure function relationships in this very, very challenging context of studying water under tension. Now, in building understanding of structure function relations in general, biologists often do invoke this proximate ultimate distinction. So, so the traditional form goes like this. So we can say, well, why do ocotillo trees have red flowers? And there are these two different answers, and one is the adaptive story. We tell a story about heritable intrapopulational variation in the context of a given selective context. So if the selected context involves hummingbird pollination, then the variants, the hypothesis will go, that have red flowers, abundant nectar, no floral scent, are the ones that will be favored and come to be represented preferentially in successive generations. So that's traditionally been called ultimate evolutionary causation. And the other is the developmental story. So why are there red flowers? We can tell a story about the anthocyanin biosynthesis pathway, how these red pigments come to be expressed in these tissues, starting from their precursor compounds and all the reactions that are involved. So this distinction actually works quite well when designing a research question. And so our title really is a shout out to other biologists regarding this sort of ongoing usefulness in biological practices. In practice, the caveat is that if you really dig down, you do see that the distinction begins to break down. And here's just one example of how it breaks down, is the way that natural selection actually works. So this is a graph here on the right from a classic paper by Per Albert. And it depicts, so it's a phenotypic space described by two variables. We have two traits, and then the dots are all the variants that we observe. So the common ones, the rare ones, the teratological ones, the ones produced in the lab, mutagenesis or other interventions, everything, every combination of these traits that we're able to observe the focal species producing. And in this example, what we see is some of the spaces are completely empty. They're just, no matter how much we perturb the system, we never see a variant, some of these trait combinations. And of the possible, some of them are rare. So the green ones here represent rare. They're possible because we observe them, but they're very rarely observed. And the blue ones, of the possible, the blue ones are common. And these turn out to be the ones that have the highest fitness or highest performance of the possible ones. So selection itself is the biasing of development, such that the favored variants out of the possible ones are preferentially produced. So yes, all of these green ones, and green are possible, but they're very rarely seen. Normally what you see in populations are configurations more or less around the favored ones. So this is just one example that when you really dig down, ontogenetic or proximate mechanisms are inseparable from selection or ultimate ones. So for these and other reasons, there's a recent and entirely correct tendency to take this distinction with a grain of salt. So with that caveat in mind, I want to show you what's happening in our field and related ones, which you'll see names like plant evolutionary ecology, functional trait ecology. These are fields in which the practitioners essentially come from ecological backgrounds and end up landing on questions that are within the scope of evolutionary biology, normally regarding adaptation. Here's just an example of a recent publication where the effort here is to try to come up with sets of traits that are readily measured that somehow represent function, however that's construed, which isn't too awfully discussed in the field. But because of where these authors start from, they don't necessarily have the background regarding the debates on these issues in evolutionary biology over the past 40 years or so, and often start rediscovering the debates or even pitfalls that have been gone over in evolutionary biology. So let me just give two examples briefly from plant hydraulics. So these authors want to explain why veins of leaves are predictably wider given leaf size. So here's a little chunk out of a leaf, and there are veins that move water from the base of the leaf, right, where the water exits the stem, enters the leaf, and water is moved up through the leaf, up to the terminal veins where it enters the leaf mesophyll and then exits the stomata. And these veins actually they're maximally narrow where the water enters the mesophyll, and they become wider and wider and wider as you go toward the base of the leaf. And they do this quite predictably. If you look at, so here's this little graph at upper right from this publication here, this Sack et al. Scaling of leaf venation architecture. If you measure the vein diameter and plot that against leaf area across species, so this is across a whole bunch of species, you come up with this very predictable allometric relationship. So they wanted to explain why big leaves have wide veins and little leaves have narrow veins, and why it should be so predictable. And they present the pattern as having what they say here, global scale impacts on leaf ecology, plant carbon economy, drought tolerance, the history of vegetation worldwide. So they're appealing to the ultimate causation, so these effects of selection. And in their efforts to explain the causes of this structure-function correlation, they appealed to proximate causes. So what they did was they proposed a developmental model based on gene expressions and hormonal signaling. I think from Arabidopsis. And they suggest that developmental causes explain then this scaling, this alignment of vein size and leaf size. And they conclude that plants have no other choice developmentally but to produce these patterns. That is no other alternatives. They do successfully predict observed patterns of leaf venation, but they infer then that there are no other alternatives possible. But plants can, in their natural variation or an induced variation, produce vastly differing venation patterns from the commonly observed ones. So these are ones from the lab of Enrico Scarpella, which he has been working on in Arabidopsis, which is very striking. So the white, so these ghostly outlines of the leaves, and the white is the venation. You can see here, it's entirely possible to produce a leaf. This is the base of the leaf here. It's entirely possible to produce a leaf that does not have the predicted wide veins at the base. In fact, this one doesn't even have veins. This upper one does not even have veins at the base. So that is entirely possible developmentally. This one here, as well, the veins are wider and become narrower toward the base of the leaf. Definitely not what would be predicted given this developmental model. So the universe of developmental possibilities is very wide, vastly wider than that depicted in the model, which only predicts that one configuration favored by selection. In fact, that, or rather predicting the common one. And that common one is precisely the one that minimizes hydraulic resistance and therefore maximizes photosynthesis as leaves get larger. This is shown by an optimality model and some functional studies. So this is what I mean by this preferential representation of the variant with the highest performance among the possible as natural selection. So this example where we start with an interest in a structure-function relationship and end up appealing to developmental causes exclusively to explain them, highlights the way that the proximate-ultimate distinction in practice helps remind us of the need to test whether or not non-observed trait values are developmentally possible and how their performance compares to the common variants, and highlights this ongoing debate about what constitutes an acceptable explanation of the distribution of organismal traits. And I'll just give you another example. It's very similar, provided by hypotheses about why trees grow to a certain height and no more. This is perhaps, this Koch et al. Is one of the most prominent hypotheses explaining this tendency. If you go from community to community, plant community to plant community, the minimum plant sizes are about the same, but the maxima are very different. And so in any given site, there will be a maximum tree height, given the microsite characteristics. And trees will grow to that height and stay there, sometimes for centuries. And in globally, you can move from areas of low to high water availability, and in certain sites in the world, trees reach their global maximum of about 120 meters or so. And this hypothesis posits for this height limitation a purely passive, or invokes proximate causes in which accumulating hydraulic resistance, so as trees get taller, the distance along which water has to be conducted becomes longer, and so you get more opportunity for friction imposed by the conduit walls. So there's more hydraulic resistance as trees get longer and the conductive path length gets longer. Plus gravity, there's this cumulative effect of gravity as trees get taller and taller. As in this, this is a giant, this is a coast redwood here on the right, with one of our collaborators there, climbing up to its 100-meter apex. And these cumulative effects of conductive path length and gravity create developmental conditions such that plants simply cannot expand their cells sufficiently to grow any taller. So again, what they don't test is whether there are in fact plants are developmentally capable of producing wider cells in trees at their height limits. But whether these variants with wider cells and potentially being taller would have lower performance and fitness than the commonly observed variants that stop at a certain height. So again, as part of day-to-day biological practice, this distinction, this proximate-ultimate distinction as traditionally construed, helps keep this need to study developmental variation and compare performance between variants front and center, rather than simply assuming what we see commonly are the only developmental possibilities. So this brings us to what we could call evo-devo adaptationism or enlightened post-spandrels, evo-devo adaptationism. It takes as an object of study not just the filled areas of phenotypic space, but also the empty ones. So so we'll take, in this diagram, we have this relationship between two traits, as this allometric relationship here. The observed data are the points. And the questions we can ask don't just have to do with the filled space, but the empty space. So we can ask, are these empty space morphologies produced in development within populations of the focal species? So can we just go and look at standing intrapopulational variation and see if maybe in seedlings or something, these variants are produced? And if they can be produced, are they common? Are they being produced all the time in equal frequencies with the common one? In seedlings, for example, are they rare? Are they only observed in early ontogenetic stages? Are they teratological? If the variation incurring into these empty zones is limited, we can look at other species. We can ask if other species, other than those examined, produce these morphologies. At least we know that organismal developmental systems are capable of making those combinations. We can try to drag populations here under artificial selection or other alterations to ontogeny, right? So we could start with a wolf and end up with Chihuahua and see that trait combination is, for better or worse, possible within that species. We can also ask if these morphologies can be produced. So either naturally or by selection or induced mutation or even by surgical intervention. We can compare their performance to the commonly observed ones, and we can see if their performance is higher or lower. So if we find that the fitness of the empty space or variance is lower, then this is consistent with selection favoring a subset of the possible. But if it's higher, then that's very strange. Why aren't those the common variants? Or if the spaces are apparently inaccessible developmentally, why is that so? So just by way of conclusion, we'd just like to point out that as a matter of biological practice, this traditional proximate-ultimate distinction does seem like a useful way of reminding us that in our search to construct explanations for organismal trait distributions, we need to explore the space of developmental possibility, not just the commonly occupied space. So this insight, this need to examine developmental possibility is maybe the main fruit of the debate over adaptationism, certainly over the last 40 years. So fields like plant evolutionary ecology and plant hydraulics show that this debate over what causes trait distributions is not by any means over, but it's playing out over and over and over again in current biology. The role of development in studies of adaptation is just one example, but there are many others, right? So, such as what do we mean? What do biologists or scientists construe as function? What do they mean when they talk about function in a hydraulic xylem hydraulic study, for example? Ad hoc adaptationist explanations propping up favorite hypotheses. It's still going on. It doesn't seem like there's much literature anymore, at least recently, in the philosophical literature on the debate over adaptationism, but certainly does seem to still be going on. What philosophical questions are contained in that, I guess, is for philosophers to decide, but it's useful to know that the debate is by no means over. Debate over adaptationism, it's very much a living thing. Not necessarily as a conscious debate as in evolutionary biology, but certainly with an expression in other fields. And for our concern as working biologists wanting to improve practice in our field, we think that this traditional proximate ultimate distinction can provide a help for workers to engage with these conceptual tools developed in evolutionary biology over the past 40 years, especially this need to examine developmental potential and the relative performance of common, rare, teratological, and imaginable in efforts to explain the vast space of organismal form. So I will stop there, and I will thank, like to thank Özlem and John for their organization, and I'd like to thank my co-authors very much for these long discussions.